Hello dear students, today we're going to talk about cancer genetics and the lecture will be entitled Molecular Biology of Cancer. The objectives that will be covered in this lecture will be to define oncogenes as positive regulators of cancer, describe how oncogenes cause cancer, define tumor suppressor genes as negative regulators of cancer, describe cell cycle and checkpoints, and finally define apoptosis. So, in order to understand the biology of cancer, we first should know the definition of cancer. What is cancer? Cancer is defined as the loss of cell division control. And so, cancer cell is a cell that grows out of control. Unlike normal cells, cancer cells ignore signals to stop dividing, to specialize, or to die and to be shed. Growing in an uncontrollable manner and unable to recognize its own natural boundary, the cancer cells may spread to areas of the body where they do not belong. In a cancer cell, several genes change. بمعنى آخر, they are mutant, and the cell becomes defective. There are two types of gene mutation. One type, which is the dominant mutation, is caused by an abnormality in one gene in a pair. An example is a mutant gene that produces a defective protein that causes the growth factor receptor on a cell surface to be constantly on. And so there is no growth factor is present. The result is that the cell receives a constant message to divide. The dominant gain of function gene is often called oncogene. So oncogene, when there is a turn on, بمعنى آخر إنه إحنا يعني الخلية will be divided without any request to any growth factor. لأنه the growth factor receptor is constantly on. سواء كان موجود الليجند أو سواء كان ما موجود الليجند. The second general type of mutation is a recessive mutation and is characterized by both genes in the pair being damaged. For example, a normal gene called p53 produces a protein that turns off the cell cycle and thus helps to control cell growth. The primary function of p53 gene is to repair or destroy defective cells, thereby controlling potential cancerous cells. إذا هذا صار بmutation, this gene is called an anti-oncogene or tumor suppressor gene. فإذا هذا صار بmutation معناتها يعني الoncogene will be active all the time. This type of قلنا الجين is called an anti-oncogene or ما يسمى بالtumor suppressor gene. And if only one p53 gene in the pair is mutant, the other gene will still be able to control the cell. لأنه مثل ما قلنا it's recessive.
It's 21 hours.
However, if both genes are mutant, the off switch is lost. هذا الtumor suppressor gene اللي هو الoff switch مالت cell cycle will be lost, and the cell division is no longer under control. فبالتالي راح تدخل إلى continuous cell division. Abnormal cell division can occur either when the active genes اللي هي oncogenes are expressed or when tumor suppressor genes are lost. So in order بمعنى آخر, in order to cancer to occur, أن mutation بالtumor suppressor gene اللي هي تسوي لنا off للcell division. وهذه الحالة لازم يكون عندنا two genes are suppressed or يكون عندنا oncogenes are turned on any genes اللي هي أي أحد من الجينات لأنه هو هذا dominant mutation يسوي لنا turn on الريسبتور مالت الجروث فاكتور, فبالتالي ما تحتاج الخلية إلى أي signal to initiate division, and so it will go to a vicious circle. بمعنى آخر, in summary, cancer cells have defective in normal cellular function that allow them to divide, invade the surrounding tissue, and spread by way of vascular and or lymphatic system. These defects are the results of gene mutations, sometimes caused by even infectious viruses. It has been determined that it takes 5 to 6 mutations in one cell to cause cancer to occur. نيجي هسه على الأونكوجينز. Let's come to the oncogenes and يعني define them more precisely. An oncogene is a modified gene or a set of nucleotides that codes for a protein and it's believed to cause cancer. Genetic mutation resulting in the activation of oncogenes increase the chance that a normal cell will develop into a tumor cell. Since the 70s, dozens of oncogenes have been identified in human cancer, and it's a huge and fertile area to research in. Oncogenes are thought to be in a perpetual tug of war with tumor suppressor gene. يعني دائما عندنا هي مثل عراك مع الtumor suppressor gene which act to prevent DNA damage and keep the cell activities under control. There is much evidence to support the notion that loss of tumor suppressor gene or gain of oncogene can lead to cancer. إذن معناتها الأونكوجين إذا صار بها إذا أكو ميوتيشن ويسبب لنا activation of the oncogene رح يسبب كانسر. زين شلون يصير عندنا activation الأونكوجين؟ الأونكوجين هي presented أصلا موجودة يعني موجودة بكل ال DNA molecule بكل أنواع الكروموسومات مالتنا as ما يسمى بال proto-oncogene. البروتو-oncogene هذا النورمال جين قبل ما يصير بيه mutation is a normal gene that can become an oncogene due to mutation or increased expression. Proto-oncogene code for proteins that help to regulate cell growth and differentiation. Proto-oncogenes are often involved in signal transduction and execution of mitogenic signals, usually through their protein products. Upon activation, the proto-oncogene or its product become a tumor-inducing agent or an oncogene. Example of proto-oncogenes include RAS, WNT, MYC, and ERK. هذه هي أنواعها. بس هي شنو اللي يسبب لنا الactivation؟ الactivation can become the proto-oncogene can become an oncogene by relatively small modifications of its original function, and there are three basic activation types. شنو هي هاي الactivation types؟ Even the activation of the pro-oncogene can become an oncogene by a relatively small modification of its original function, and هذه أكو ثلاث حالات: either mutation or increase in protein concentration or chromosomal translocation. فبالmutation شي يصير عندنا mutation within the proto-oncogene can cause a change in the protein structure اللي هي that will be expressed by this protein, and this will cause an increase in protein enzyme or loss of regulation. يعني بالعكس either increase بالactivity enzyme activity or بالعكس inactivation of the enzyme. Second, an increase in protein concentration caused by either an increase of protein expression or increase in protein stability, prolonging its existence and thus its activity in the cell, or a gene duplication, one type of chromosome abnormality, هذه واحدة من عندهم, resulting in an increased amount of protein in the cell. السبب الثالث قد يكون chromosomal translocation, another type of chromosome abnormality causing an increase in gene expression in the wrong cell type, or expression of the constantly active hybrid protein. This type of aberration in a dividing stem cell in the bone marrow leads to adult leukemia. This is an example. شافوا recently mutation in microRNAs can lead to activation of oncogene. هذا سبب آخر. New research indicates that small RNAs اللي هي بين 21-25 nucleotides in length called microRNAs can control expression of these genes by downregulating them. هذه فد معلومة قد تفيد بالبحوث أو research projects مالتكم. نجي على examples that have been found examples of oncogene examples of oncogene that have been found work in tissues. عندنا السيس، عندنا الراس، عندنا الاربي، عندنا السيرك، عندنا الميك. السيس اللي هو سيميا ساركوما and الراس اللي هو رات ساركوما effects of the G protein. الاربي هذه بالchicken affect growth receptor sites erythroblastosis. And السيرك اللي هو rose sarcoma protein kinases called causes signals in the S phase يعني بالsynthesis مالت DNA phase. والميك اللي هو myoplastic affects white blood cells and leukemia. أول oncogene discovered بالسبعينات in the 70 كان هو السيرك pronounced SARC. اللي SARC was in fact first discovered as an oncogene in the chicken retrovirus. وبعدين بال 1976 doctors Michael Bishop and Harold Vermes of the University of California, San Francisco demonstrated that oncogenes were defective proto-oncogenes. يعني هم اللي اكتشفوا إنه الأونكوجينز هي بالحقيقة موجودة بس لكن in inactive form سموها proto-oncogenes، وهذه يصير بها defective يصير بها defect يلا يصير عندنا oncogene. وهذه الديسكفري then were يعني من هذا الديسكفري were awarded a Nobel Prize بال 1989. زين هسه how do oncogenes cause cancer? The change of an oncogene from normal to cancerous function can be caused by a single point mutation in the sequence of a gene. خلينا ناخذ for example مثلا a change in the RAS oncogene located on the human chromosome 11 from guanine to cytosine. يعني هو كان guanine بالبروتو oncogene تحول إلى cytosine يعني this is the product مالته frequently associated with bladder cancer. This simple change results in glycine at amino acid 12 being substituted with valine. This dramatically change يعني هو فقط هذا البروتين that is expressed by الأونكوجين هذا هو التغير اللي صار به. This dramatically changes the function of the G protein encoded by the RAS gene. Normally the protein cycles from an inactive to active state by change the bounding مالت GDP to GTP. The mutation does not allow the release of GTP and the protein is constantly active because the signal delivered by the RAS oncoprotein is continuously delivered. The cell continues to grow and divide, and this will enable the growth matter and leads to it will enable continuous growth and leads to bladder cancer. إذا هنا عندنا هذا الإكزامبل مالتنا هو الراس بروتو أونكوجين. mutation in the RAS gene are found in about 30% of human cancer. يعني كل شوية يسبب الهيومن كانسر. والبرودكت مالته هو يأثر على الجي بروتين. هذا الجي بروتين that relays a growth signal from a growth factor receptor يعني فبالتالي on the plasma membrane to a cascade of protein kinases. فما ما نحتاج ذاك الوقت ligand. This receptor is always activated leading to continuous cell growth. نجي هسه للtumor suppressor genes اللي هي negative regulators للكانسر. هذه some genes suppresses tumor formation بالحالات الطبيعية بس إذا صار بها mutation لا صار عندنا مشكلة. Their protein products inhibit mitosis. When mutant هذه الجينات الميوتنت allele behaves as a recessive، يعني لازم عندنا التو alleles لازم يكون بيها mutation، يلا يعني راح يصير mutation للجين. Tumor suppressor continues. الoncogenes by contrast behaves as dominant، يعني one mutant or overly active allele can predispose the cell to tumor formation. يعني الtumor suppressor genes لازم التو alleles يصير بيها mutation، يلا يتوقف العمل مالتها، بينما الأونكوجينز يعني dominant، واحد يشتغل يسوي mutation للبروتو أونكوجين يسوي لنا أونكوجين. نجي هسه على الإكزامبل. أول إكزامبل هو الريتينوبلاستوما جين. Retinoblastoma gene is a juvenile eye cancer that is caused by a mutation in the retinoblastoma gene located on chromosome 13 of humans. هذا الجين suppresses the development of cancer at its dominant phenotype مثل ما قلنا. Therefore, both alleles must be mutant for the disease to develop. فأن this is هذه هي healthy eye، هاي retinoblastoma على الرتينة as you could see من اسمها retinoblastoma. This is the healthy eye، وهذه retinoblastoma eye اللي هي cancerous cells. هذه cancerous cells ما تحصل إذا كان عندنا، لأن هو يعني عندنا التيومر سبريسر جين عندنا اللي هو ريتينوبلاستوما. This is a tumor suppressor gene. إذا صار بي mutation، وهذا الميوتيشن لازم يكون عندنا بالboth alleles، يعني both parents لازم عندهم هالميوتيشن، يلا يصير عنده بالطفل الكانسرس cell. How do الريتينوبلاستوما gene activate ال S phase؟ إن ال S phase is activated as you can see here by the E2 factor. E2 factor is when the E2 factor is released اللي حيسوي لنا activation stimulate the production of the S phase proteins and this will enter. فبالتالي ال S phase هو الفيز اللي بالسايكل اللي حيصير بها synthesis of DNA، فبالتالي راح يصير عندنا الاكتيفيشن وتدخل الى سيل سايكل جديده الخليه. فاذا منو اللي يسوي لنا اكتيفيشن للاس فيز؟ هو الاي 2 فاكتور. الاي 2 فاكتور هذا is released only اذا كان عندنا الار بي is phosphorylated. الار بي product is phosphorylated. الار بي product بالنورمال حالات النورمال is not phosphorylated by kinases. فبالتالي when normal cells ال RB proteins binds to E2F اللي هو يعني growth factor and so it will shut down the cell cycle لأنه هنا عندنا ال E2 is not available to activate ال S phase، يعني ال RB لازمة. بينما إذا ال RB صار ب phosphorylation by the kinases ال E2 will be released and this will cause ال S phase activation. شو وقت يصير عندنا phosphorylation لل RB الريتينوبلاستوما جين؟ لما يصير عندنا mutation بهذا الجين. Retinoblastoma is a cancerous tumor of the retina. It occurs in two forms. أكو familial retinoblastoma اللي هي multiple tumors in the retinas of both eyes occurring in the first week of infancy. وأكو sporadic. A single tumor appears in one eye sometimes in early childhood before the retina is fully developed and mitosis in its ceases. بالfamilial retinoblastoma occurs when the fetus inherits from one of its parents' chromosome number 13 that has its locus deleted or يعني mutant والnormal RB proteins prevents mitosis. الميكانيزم هنا ال RB protein prevents cells from entering ال S phase مثل ما قلنا. it does this by binding to the transcription factor اللي هو ال E2 اللي هو احنا نسميه يعني initiation factor 2 and فبالتالي ال E2 forms binding to the promoters of such proto-oncogenes as ال c-myc and c-fos. هذه ما علينا، أنا ما نريد ندخل في تفاصيلها، وبالتالي راح يسبب لنا مثل ما قلنا activation of the S phase. فهذه هي الحالة. عندنا بالنورمال كروموسوم هذه قلنا بالنورمال normal cell normal retinal cell ان two alleles are normal بينما هنا deletion is يعني بالنمبر 13 and this will cause اللوكس فبالتالي this will cause RB protein. And بالsporadic in this disease, both inherited RB genes are normal and a single cell must be so unlucky as suffers of a sporadic mutation, often a deletion in both in order to develop into a tumor. Such a double hit is an exceedingly probable event, and so only rarely will such a tumor occur. In both forms of disease, the parent life can be saved if patient's life can be saved if the tumor is detected soon enough and the effect of the affected eye is removed. Highly sporadic. الإكزامبل الثاني للtumor suppressor gene is the P53 and the product of the tumor suppressor gene is a protein of 53 kilodalton من هذا هو اسمه P53 located بالchromosome 17 and this protein contains 339 amino acid and a single amino acid substitution can lead to loss of function of the gene. Mutation at amino acids location 175, 248 and 273 can lead to loss of function and changes at 273 تقريبا 13% are the most common. These all act as recessive mutation. Because these mutations can be expressed in heterozygous condition, they are often associated with cancers. This genetic function of the gene of this gene is to prevent cell division of cells with damaged DNA. Damaged DNA could contain genetic changes that promote uncontrolled cell growth, therefore preventing cell division until damaged DNA is repaired. هذا هو main function مالت ال P53. يعني P53 هو يمنع الدامج سيل انه to divide الى ان يصير بيها repair يلا يصير بيها division. فاذا ال P53 gene is suppressor gene is mutant فبالتالي ما راح تتوقف السيل division اذا كان عندنا damage بالدي ان اي وبالتالي السيل will be divided in spite of the damage of the DNA before its repair. These functions make P53 a key player in the protecting against cancer. That is important tumor suppressor gene. هذا واحد من أهم الجينات اللي هي يعني الموجودة في الجسم وأيضا يعني it's a fertile area for research and in cancer research. Mice have been cured of cancer, been treating them with peptide that turns on production of the P53 protein in the tumor cells. However, excess production of P53 protein leads to accelerated aging in mice. However, under more physiological conditions, P53 seem to protect against both cancer and aging, perhaps by protecting cells from oxidative damage, as you can see from this figure. نلاحظ بهذه الصورة هي الفانكشن مالت ال P53 شلون يشتغل ال P53 بروتين شلون يشتغل إذا صار عندنا دامج بالدي ان اي رأسا تنهانسز الدي ان اي ريبير انزيم وبالتالي تمشي السيل سايكل with normal DNA. بينما إذا كان أكو دامج وال P53 is mutant فبالتالي البرودكت مالته will be non-functioning and so the cell will be divided in spite of its damaged DNA and this will lead to production of damaged cells, damaged DNA cells and a progression to cancer cells. النوع الثالث أو الإكزامبل الثالث هو الفاب اللي هو familial adenopolyposis in the intestine which are precancerous. المثال الأخير هو P16 وهذا يشبه P53. البرودكت مالته هو بروتين which is 16 كيلو دالتون فمن هذا هو اسمه. أيضا كلش يشبه ال P53. In humans, deletion and other mutations of P16 are found in variety of cancer. نجي هسه على loss of heterozygosity. Because the tumor suppressor genes are recessive cells that contain one normal and one mutant gene that is are يعني heterozygous still behave normally. يعني بمعنى آخر الهيتروزايغوس يعني ما تأثر على الفانكشن مالت الجين product. However, there are several mechanisms which can cause a cell to lose its normal gene and thus be predisposed to develop into a tumor. This results in loss of heterozygosity. يعني حتى لو heterozygous قد يؤدي إلى abnormal products. شنو هي الميكانيزم of loss of heterozygosity؟ قلنا الميكانيزم of loss of heterozygosity deletion of the normal allele or chromosome arm containing the normal allele, or in females the S inactivation of the S chromosome carrying the normal allele, loss of the chromosome containing the normal allele, mitotic recombination اللي هو the study of tumor suppressor gene revealed for the first time that crossing over with genetic recombination occasionally occurs in the mitosis. And بالحالات الثالثة والرابعة the resulting cell now carries 2 copies of the bad gene, and فبالتالي this is called the reduction of homozygosity. A mutation is not the only way to inactivate a tumor suppressor gene. يعني tumor suppressor gene is not only activated by mutation. Their function can be blocked or lost by methylation. Cancer cells often contain methylated promoters للtumor suppressor gene. Loss of the locus of the other chromosome اللي هي مثل ما عندنا بالloss of heterozygosity بالنقطة الأولى, و inactivating mutation in the other allele. الفيروسز human papilloma virus, the name of the anti-oncogene may be even more appropriate than originally thought. Both retinoblastoma protein and p53 protein turn out to complex directly in the cell with an oncogene product of some human papilloma viruses. وبالتالي هذه تسبب لنا أيضا نوع من promoters and that promote cell growth and cell signaling. Inside the cells of their host هاي papilloma viruses synthesize a protein اللي هو E7 مثل initiation factor, وهذا ال E7 binds to the retinoblastoma protein preventing it from binding to the host transcriptional factor E2. وهذا أيضا يعني شلون إحنا بالحالات العادية هو المفروض يتصل بال E2 normal cases normal cells يتصل بال E2 على to prevent it from activating of the S phase. هنا عندنا بالحالات الغير عادية إذا صار بها phosphorylation للريتينوبلاستوما جين راح يصير عندنا ما راح يقدر يتصل بال E2. هنا عندنا هذا ال E7 اللي هو ال product of the human papilloma virus كأنه مثل يسوي phosphorylation. هو مو يسوي phosphorylation بس يعني كأنه نفس الميكانيزم مال phosphorylation of the retinoblastoma virus, وبالتالي ما راح يقدر يتصل بالE2, وبالتالي الخلية راح will be activated by E2 will be available to activate the S phase بالcell cycle. نجي هسه على شنو هي ال S cycle. To ensure the proper performance of each organ يعني مثلا for example any cell that is worn out that is injured must be replaced, وهذه replacement is done by a cell cycle. cell cycle هي عملية تصنيع خلايا جديدة و proliferated new cells by طبعا multiple phases من ضمنها ال S phase اللي هي synthesis of the DNA يعني DNA molecule. examples إني شو وقت نحتاج إنه يصير عندنا proliferation للcells بالbone marrow increase its production of oxygen carrying RBCs. Certain white BCs are produced more rapidly during infection for example, فنحتاج cell cycle. liver and endocrine organs frequently respond to injury by regenerating damaged cells. Reproduction of cells is a process of cell division, and the division of normal cells is a highly regulated process. The cell growth, inheritance, and containment of its controlled by its DNA deoxyribonucleic acid. cell divide only when they receive the proper signals from growth factors. هذه أهم شيء that اللي إنه عندنا cell cycle is controlled الphases مالت ال cycle is controlled by checkpoints, and these checkpoints are the growth factors, and فبالتالي that circulate in the bloodstream or from a cell that directly is in contact with it. When a cell receives a message to divide, it goes through the cell cycle, which includes several phases of the division to be completed. Checkpoints along each step of the process make sure that everything goes the way that it should be. هذه هي الفيزز مالت السيل سايكل اللي هي جي نوت والاس جي نوت وجي 1 جي 2 اس فيز اند الميتوسيس. وطبعا هذه الحمر هي التشيك بوينتس اللي هي الريجوليتوري فاكتورز اللي مثل شلون نقاط سيطره اللي لازم ما تدخل السيل سايكل، ما تدخل الخليه الى الفيز مالت السيل سايكل الا لما تتاكد من الريكوايرمنتس. التشيك بوينتس هذه هو ياكد لنا اياها. شنو هي التشيك بوينتس؟ بالفيز الاولى التشيك بوينت فيرست جاب، التشيك بوينت اللي هي الاول تشيك بوينت اللي هي لازم عندنا السيل سايز is adequate, nutrients is available and growth factors are also available and present. التشيك بوينت الثانيه اللي هي بالجي نوت يعني second gap، عندنا هم نفس الشيء cell size is adequate, chromosome replication is successfully completed. يلا تدخل الى الجي 2 الثانيه. الميتا فيز اللي هي check point pass check point if the all chromosomes are attached to mitotic spindle، وهذه تحصل اذا صار عندنا ال DNA synthesis was well done. Availability مالت growth factors for example which are involved in passing of the G1 checkpoint. هذه طبعا هاي ال growth factors are presented in the cell and يسبب لنا زيادة بما يسمى بالcycline concentration. والcycline activates cycline dependent kinases، وبالتالي هذه هاي cycline dependent kinases راح تتصل بالtarget protein and activates ال S phase بالcell leading to cell division. طبعا of course الأهم شيء يعني بالcell division هي ال S phase لأنه يصير عندنا يصير عندنا entrance to the S phase، يصير عندنا synthesis مالت ال DNA. الregulation مالت ال cell cycle checkpoints occur at key transitions in the cell cycle and provide go or no go decision point بالضبط مثل السيطرة. It is now known that sequential activation and inactivation of the cycline dependent kinases هي هذه أهم شيء is the primary means of the cell cycle regulation. Thus this is another example of the importance of phosphorylation dephosphorylation in biochemistry of cell regulation. Alterations of one or more of these checkpoints control occurs in most of it if not all human cancers at some stage in their progression to invasive cancer. يعني كلش مهم نفتهم التشيك بوينتس والافيلابيلتي مالت السايكلين كاينيزز سايكلين ديبندنت كاينيزز والافيلابيلتي مالت الجروث فاكتور على مود نفتهم البروجريشن initiation and progression of cancer cells. Examples of these alteration, for example, alteration of the G1 S checkpoint occurs in many humans. For example, هذه هي key player بهذه checkpoint is the retinoblastoma gene. هذا كلش مهم. Phosphorylation of the retinoblastoma راح يسبب لنا cyclin D dependent kinase release, and this will enter the S phase. Inactivation of the RB by genetic alteration occurs in retinoblastoma and is also observed in other human cancers, for example, small cell lung carcinoma and osteogenetic sarcomas. P53 gene product هذه همين أخذنا مثال عليها important in the cell cycle checkpoint regulation بالboth. هنا الجين ال P53 regulates both checkpoints بالG0S والG2M checkpoints. Tumor cells lacking normal p53 function do not arrest G نوت G1 I'm sorry. فبالتالي ما عندنا احنا G نوت، ما عندنا senescence space، فبالتالي رح تدخل إلى cell cycle جديدة من خلال activation of the S phase مرة ثانية. Apoptosis, sometimes called programmed cell death, is a cell suicide mechanism that enables multicellular organisms to regulate cell number in tissues and to eliminate unneeded or aging cells as an organism develops. The biochemistry of apoptosis has been well studied in recent years, and the mechanism are now reasonably well understood. يمكن أفضل لو تشوفون فيديوهات على اليوتيوب concerning الأبوبتوسيس والسيل سايكل، يمكن تصير أوضح للفهم مالتكم. هذه هي الميكانيزم of apoptosis، الكاسبيس dependent والكاسبيس independent. ان الكاسبيس dependent هي الانترنسيك فاكتور أو اكو الاكسترنسيك فاكتورز. The genes involved as players in the apoptosis include MYC, P53, FOS, and interleukin 1 beta converting enzyme اللي هو ICE. Various oncogenes product can suppress apoptosis. These include adenovirus protein, RAS, and V-ABL. Where apoptosis occurs in most of, if not all, solid cancers. For example, يعني هاي هي الأمثلة dividing cells and midline non-dividing cells والcore hypoxic.
It's 22:30.
It would be therapeutically advantageous to tip the balance in favor of apoptosis over mitosis. يعني هاي تلاحظون إنه دائما عندنا فد balance between them. Programmed cell death against mitosis اللي هو cell division. لازم اثنيناتهم يكونون على شو اسمه على ميزان واحد. يعني it is clear that number of anti-cancer drugs induces apoptosis in cancerous cells. You know, apoptosis only occurs in normal healthy cells. The problem is that they usually do this in normal proliferating cells. Therefore, the goal should be to manipulate selectively the gene involved in inducing apoptosis in tumor cells. Understanding how these genes work may go a long way in achieving this goal. هذه هي therapeutic يعني application of the medical importance of caspases and the therapeutic activation of apoptosis or importance of apoptosis. And thank you very much.